All right, so in chapter 36, I'm going to go through some of the things with you, and it deals with xylem and phloem, and that the structure and function of xylem and phloem in plants are correlated with one another. So some of the things I'm going to go through, and some of the things I'm just going to kind of let you look at because we've talked about them in the past with water potential and things like that. So with chapter 36, with solute movement, the plant's plasma membrane, just like all membranes, is selectively permeable, and it's going to regulate what moves in and out of the cell. And it has some active processes where ATP is used, and it has some passive processes where it's just going to move according to really the difference in a concentration gradient. And transport proteins, if you recall, these transmembrane proteins, these channel proteins, these things that allow things to move in and out of the cell are what are going to be involved. So with active transport, you've got a bunch of proton pumps, and these really in plants are what's going to be really important because they're going to use ATP to build up a concentration gradient of these hydrogen ions, and then as a result of that, you're going to be able to move things back and forth across the membrane due to the potential energy gradient from these hydrogen ions. And you're going to have a little bit of an electrical charge difference too, so that's going to help the cell be able to do this work as well. So plants use these hydrogen gradients, as we just said. So if you look here on the bottom left figure, you've got some ATP being burned, and that's used to drive these hydrogen ions across the membrane. And then to kind of balance the charge, these potassium ions can move in, and we'll see how this is going to regulate the opening and closing of the stomata on the plant cells and things like that. In the root cells, we can see here the potassium gradient is going to be used to allow water to be taken up by the cell or by the organism, however you want to look at it. So it's a pretty interesting process. So the idea of co-transport here, this is an active process, so that's one of the important things that you're going to want to remember is co-transport is an active process. But what's happening here is you've got the downhill flow of one solute kind of coupled with the uphill movement of another. So as we just mentioned, we're using some ATP to generate a hydrogen ion gradient. And then with that gradient now, these hydrogen ions want to move back into the cell where they came from. So as they move in, they're going to bring sucrose in this example, because the plants are going to be making sugar, and sucrose is an example of a sugar that plants like sugarcane, things like that make. As these hydrogen ions move back across the membrane, these sucrose molecules are going to come with it. So that's co-transport. Now, if you recall with osmosis, this is just a passive movement of water. It's the diffusion of water from a high concentration of water where water is pure or more pure to an area where it's lower in concentration. So the water is going to move to where the solutes are, more or less. And this uptake or loss of water is what's going to be vital to a plant's survival. So there's water as it's moving through. There's two various pathways. There's apoplastic, where it's kind of between the cell outside and inside the cell, and then the symplastic, which is connected through these plasmodesmata. So all the cytoplasms are together, and you can see that the symplastic route is going to kind of move through this cytoplasm, whereas the apoplastic route is in the red there. That's going to move on the kind of the outside of the cell membrane through the cell walls and such. So with osmosis, if these plant cells are impermeable to these solutes, then if you know the concentration of either side of the membrane, then you're going to know how the water is going to move. And this is some of the stuff that I'm going to just let you kind of look at as I go through. You can pause it and kind of read it and just recall what we talked about, like when we did the potato lab and things like that, dealing with water potential and solute potential and those sorts of things. And certainly if you have any questions, then we'll be able to go over these things in class. But just, if you're putting a cell into a concentrated solution, if the surroundings are hypertonic compared to the cell's going to lose water to the surroundings. And if you put it in a situation or a beaker, a container that has pure water, let's say, so now the inside of the cell has a much higher concentration of solutes than the outside, then the water is going to rush in. And that's kind of what we saw when we did the potato lab. And then the free water, that's what's not bound to any solute. So anytime you put a solute in the water, you're going to bind these water particles to it. And the water potential now is going to go down. So water potential is typically measured in megapascals. Sometimes you see it in bars. And those are just the the two general kind of units you're going to want to put on your thing if it's talking about pressure. Now, this water potential that we discussed, we always said that pure water, just by definition, has a water potential of 0. So it's not bound to anything, and anytime you start adding a solute potential to the, or excuse me, a solute to the water, you're going to affect the water potential. And so you've got your equation there. The water potential is kind of the sum of the solute potential as well as the pressure potential. And so again, if you have any sort of a concentrated solution, your water potential is going to be negative. Okay, so you're going to have less water available when solutes are in there because some of it's going to be bound, so that's why it would be negative. So if you just kind of look through and recall some of these things that we talked about in the past, I'd recommend pausing it, just kind of looking at it, reading over it, and certainly if you have any questions, make sure that you write them down, you ask about them, and we can definitely go over them in class. This is stuff we covered, I would say last year, probably somewhere around chapter 7, chapter 8. So now as far as transpiration has occurred, what transpiration is the loss of water from a plant through the stomata and the leaf. And this is just a natural consequence of gas exchange in the plant. The plant needs some oxygen. It also needs CO2. It's got some water it's going to be getting rid of. So as these things are kind of exchanged, water is just lost as the process. And if you look at the leaf anatomy, the insides of the leaf are specialized by according to their function. So the upper side of the leaf has lots and lots of chloroplasts on it. It's got that palisade mesophyll. It's very closely packed together, very dense, and the underside of it is the spongy mesophyll. There's lots and lots of internal surface area that increases in these spaces, the surface area by 10 to 30 times, and that's primed for the gas exchange. And we'll see a couple of kind of pictures of what this might look like later on. So this large internal leaf anatomy, not only is it primed for gas exchange, but you get a lot of evaporative loss of water from that as well. And these stomata and guard cells that are associated with the stomata, these teeny mouths, I guess you'd say, on the underside of the leaf, more so than on the top of the leaf. This is what helped to kind of exchange the water loss, the gas exchange, that sort of a thing. And the guard cells are trying to balance the water loss with the photosynthetic requirements. So on hot, windy, sunny days, this is where you see lots and lots of transpiration. You get a lot of evaporative water loss. The plants might wilt. And one of the benefits to this water loss is as the water is leaving, so too is some of the heat. So this is one way that the plants can kind of stay cool and make sure that the enzymes that are making up their photosynthetic systems are not being disrupted in any way. So now these stomata of the plants, these things open and close due to changes in the environment. And these are they're called guard cells because they're the sentry that's going to allow these stomata to open and close and allow for the gas exchange to actually occur. And the guard cells become flaccid or turgid depending on how much water, how many ions there are in there, and how much water is available to the plant. And they want to remain closed on these hot, dry, windy days because you don't want to lose too much water to damage the plant, but at the same time they've got to meet those requirements for photosynthesis to occur. So they're trying to balance those two things. So if you look here, you can see the guard cells on the bottom in the left figure there. They're open. There's a lot of transpiration taking place. And then on the bottom right there, you can see that they're closed up and, trying to prevent this water loss and any damage that could be caused by these plants if they start going through plasmolysis and wilt too much. So now looking at this particular kind of turgor pressure that we've talked about in the past here, you can see on the left the stomata are open, and these potassium ions, as these things are moving in, what they're doing is they're lowering the water potential. That's some of the slides that we were just going over. It was just kind of a review to build up to this point. When you have lots of solutes in the water potential gets lowered, and as a result, any of the water in the surrounding cells is going to want to kind of move in there to kind of balance the ions out as much as possible. And in doing so, they swell up, they get turgid, and they open up. And that's what's going to allow for this gas exchange. And if you do the opposite now, if the the plant wants to make sure that it's not going to lose too much water or anything like that, the ions are going to move out of the cell, and so too is the water. It's going to follow, and then these things are going to become flaccid and they're going to close, and that's what's going to prevent the massive amount of water from being lost. So the movement of these ions in and out are kind of like that co-transport thing that we saw earlier. So pumping these hydrogen ions out of the cell drives potassium into the cell to kind of balance out that charge, and as a result, that's what's going to affect the water potential. And this process is an active process, and it requires a lot of ATP, and you're going to get this from sunlight. That's going to power the production of the ATP, which is going to help with this entire process. So, that's how the plant's kind of opening and closing these things. So now there's three cues really that govern stomatal opening. And you have light, you have carbon dioxide levels, and then you have just the general circadian rhythm, that biological internal clock that's kind of related to the 24-hour day. So the light receptors, these stimulate the activation of these ATP-powered pumps that we were talking about, and this promotes the uptake of the potassium, which is going to cause the water to rush in, the stomata to cell become turgid and open up. The CO2 levels, when the CO2 levels drop, there's, the stomata are going to open because photosynthesis requires the CO2, so they're going to open up to let more in. And then just the general circadian rhythm, these things, they don't need to be open too much at night because there's no light to drive photosynthesis. So, they're going to want to remain closed. So just general circadian rhythm with this biological clock, they kind of get in cue with the light and dark cycle. So the way this applies is there's three main routes for water and solutes to move through, and we kind of talked a little bit about some of these things. And the substances can move back and forth across the plasma membrane, of course. And then once they enter the cell, they've got a variety of ways that they can move. And if they move through, just the symplast, that's all of the connected cytoplasms of these cells, they move through the plasmodesmata, and you can get short distance movement of water and things very easily that way. But the thing is if we want to do it kind of long distance, this is where we're going to have to get the xylem vessels involved. So the apoplastic route, this is just kind of the short kind of water can be absorbed, can be moved in, but you can't get it from the roots to the top of like a 300-foot tall redwood tree or something. You're going to have to involve pressure and other things. So this bolt flow, like we said, this apoplastic route is good for the short distances, but for the long distances, like the tops of these trees, this is where we're going to have to get pressure involved. And negative pressure is going to drive this long distance transport in these xylem vessels that we were discussing. This transpiration now, this is the water loss that reduces the pressure inside of the leaf. And this is going to create kind of a tension that's going to pull the xylem upward through the roots, through the trunk of the plant, and kind of, power this whole process, so to speak. And we get active transport. This is I'm going to move the ions back and forth across the cells and the roots, and that's what's going to alter the water potential in such a way that we're going to be able to pull water into the plant, and then the water is going to be pulled up the plant. If you can recall last year going over water and talking about the cohesive and the adhesive forces, those things are going to be in play. The cohesion between the water molecules pulling on one another, and then the adhesion between the water molecule and the sides of these xylem pipes moving up all the way to the top of the plant. And then in addition to that, just getting the difference in water potential between the dry air at the top of the plant and the kind of water in the leaf, the water in the branch, the water in the trunk, and then the water that's being taken up by the roots. So this water potential difference is going to be very important for moving the water up the plant as well. So once we draw the water into the plant now, we're going to increase that water potential or water pressure within the cells, and this is going to act to kind of push the water upwards. And you're also going to have this pull that I was talking about earlier, but sometimes you get this phenomenon called guttation, where you can actually see this process of this water being pushed in. And it's just kind of a little aside here to look at. This is not dew, it's actually being pushed out of the little pores there in the plant because so much water is being taken up in the roots of those plants. But back to transpiration. If you look here, you can see this little opening, these stomata, these tiny, mouths that are going to allow the things in. And then if you look at the upper side of the leaf there, you've got a little cuticle that's going to prevent the water loss. You've got those kind of columnar shaped cells. That's the palisade mesophyll, and that's where there's lots of chloroplasts. And then the spongy part is kind of from the middle of the leaf downward, and that spongy part is going to be primed for the gas exchange, and that's where you're going to have a lot of evaporative water loss. You're going to have lots of oxygen being given off from the process of photosynthesis, a lot of CO2 being taken up, and this stomatal opening is going to kind of create this pull that we've been talking about, because the dry surrounding air has a much lower water potential than the water inside the cell, so it's going to kind of pull it out, and as it pulls the water out of these stomata, kind of on the other side of that, it's going to continue to pull it up from the tissues in the tree, all the way down from the roots. So you can see this is some of the stuff that we were just talking about in terms of the water potential, the air being very dry, having a very low water potential, and that's what's going to pull the water out of the plant during the process of transpiration. And here's kind of a little bit better of a picture of it. If you look really close, you can see the gas exchange between the CO2 and the oxygen, but you also have this airspace where there's a very low water potential, and it's much lower than it is in the cells, and in the cells it's a little bit lower than it is in the associated xylem vessels and so forth. So the water is going to continually be pulled towards the outside, and you're going to have these cohesive and adhesive forces that work all the way along the length of the plant, all the way down to the roots, where those active processes are pulling water into the plant. So that's what's going to drive this whole process of transpiration. So here's a better look at it. If you're looking at the tree, you can see down in the soil, you can see what the water potential is there at the bottom. It's 0.3 megapascals. And then in the very outside dry air, you can see up at the top, it's negative 100. So we're going to flow from high to low. And that's the water potential gradient is going from being very high down in the roots to very low in the outside air. And these cohesive and adhesive forces, along with this water potential, is what's going to act to pull the water out of the plant. So these are the process of hydrogen bonding. This is a lot of the stuff that I had just mentioned to you, and you can kind of take a look at those and pause them if you need to see where the cohesive and the adhesive forces are actually at work. And then, this is the dealing with the surface tension of the transpiration and that sort of a thing. So now, as far as these xylem pipes, these tiny kind of exterior cells on the plant, they're very stiff, but they're a little bit flexible, and you get some tension that's created as this water is being pulled up the tree on the hot day. And you can actually measure these things because the walls are very kind of thick and lignified and kind of prevented from being able to collapse, but they do kind of shrink the entire trunk of the tree a little bit. You can actually measure a small change if you've got the right instrumentation in the diameter of the plant. And then occasionally what happens is in the winter, every time the water channels here, these xylem pipes freeze, they can crack, and then they get an air pocket in them, they're broke, they don't work. And then the plant's going to regenerate some more the following growing year. So when we talk about growth in plants, we'll discuss these as well. But one of the things that you can measure in terms of just how quickly certain things flow, if you, again, have the right tools, the right instruments, is on really hot days when transpiration is running really high, the xylem will move up the plant about 75 centimeters per minute. And, that's a little over 2 feet per minute. And it's about the same distance around one of our school clocks. That's about 75 centimeters. So you can see how quickly the second hand is moving. That's about how fast the xylem will move on a hot, dry, windy day. The phloem, this is what contains the organic compounds. This is the sugar. These are the things that are made during photosynthesis, and these are the things that are moving around the plant. And they always flow from the source, the leaf, to the sink, which is either the root or the fruit of the plant. And it can flow in many directions, but it's always going to flow from the leaves to one of the sink cells. And the primary source of the sugar, this is, again, usually in the leaf. Any green part of the plant is where you can perform photosynthesis. But the sink's what's going to store it. And again, like I said, roots and shoots and so forth. And the sugar transport, what you have to do is you've got to load this into these sieve tube members, and these things are going to assist with kind of the movement of these sugars throughout the plant, because you've got to be able to move them from the source, and then you got to be able to put them in a spot where the sink is. And if you think about the where they're being formed, and then how sweet a fruit might be, you can see that you're going to require some ATP in order to make this work right. So we've talked about the apoplastic and the symplastic pathway. So these sugars can move through both of these types of things, and when they do so, you need this active kind of transport mechanism to do this, because in this sieve tube member, this phloem tube here, you've got a lot, in some instances you've got a lot of sugar. So in order to get the sugar moved from a lower concentration to a higher concentration, you're going to need an active process, of course, an active transport. So the diffusion is not going to work, so you've got to actively pump these things into the sieve tube member. Now, when you're loading these things up, you've got to keep in mind too that you've got a xylem vessel that's pumping some ions and a lot of water kind of in the opposite direction. So when you have a high concentration of sugar, as you see up here near the top of this figure, the water is going to want to flow in. So in addition to this active process, you're going to get some passive processes as well that are going to assist with it, because as that water moves in, that's going to push the phloem down towards the bottom, towards that sink cell in this particular instance. And then once it gets to the sink cell, now you're going to be pumping it against a concentration gradient, so it's going to be an active process again. So you're going to have to kind of use both active and passive processes to get these things in. But now as the sugar concentration drops in that phloem tube, the ion concentration in the xylem vessel is higher, so that water is going to flow from high potential to low potential, so it's going to move back in, and that's what's going to push the xylem up. So it's almost like a cyclical type of a thing here with regards to the water. So if you look here, this is just some of the words kind of describing some of these types of things that we were just talking about. But again, you have active processes occurring to get this water, or excuse me, to get this sugar into these phloem tubes, and then to get them into the sink cells. And then water is going to assist with some of the movement of these things all along the way. And then again, here in terms of the sugar unloading, getting it to the sink, sometimes the sugar is going to be converted to an insoluble starch, but that's going to lower that water potential a little bit. Water is going to follow by osmosis and help kind of push these things in as well. And you can see that this, if you think of like a grape or a peach or something like that, it's got a lot of water as well as a lot of sugar in there. This is what's going to push these fluids in, or these processes here that we're talking about. And then this is, again, this is just some of the cycling with the water and the xylem and the phloem and how these things kind of move around. And, I just kind of went over these, some of the words to some of the stuff that I was just talking about with you. So if you have any other questions, make sure that you write those down, and we'll talk about them in class when we get back from break. Hope this helped.